Good morning. It's 9:30, and we're going to do the last part of Chapter 3. And that starts on page 103 in terms of protein synthesis, Module 3.7. You can come down and look at those learning outcomes. I'll ask you to do that at the end of the lecture. And so you see it says the process making proteins from DNA via RNA or by RNA is called protein synthesis. It's an amazing procedure. We're not going to learn everything, but we're going to learn sort of the bare bones of it. You look at the bottom of this column on page 103, the second column. You come down to the last paragraph and come down right into the middle, and you'll see these italicized words. During transcription, the code specified by a gene is copied. That's what transcription is. You copy something, creating a strand of messenger RNA. It's carrying the message of the protein. It says in translation, ribosomes read the nucleotide sequence of messenger RNA strand and synthesize a polypeptide. That would be the primary structure, not secondary, tertiary, or whatever, because we've already talked about how those structures are put together. But here we're just talking about making the polypeptide, a chain of amino acids, containing the correct amino acids in the sequence. Just like words, you got to put the right letters in the right sequence. Sometimes you get some messes on television as well as in class by not spelling things correctly. So you see down at the bottom, you got that little printout of DNA, transcription, messenger RNA, translation, and protein. So under DNA, you could put the code for a protein. Transcription takes place in the nucleus. The product of transcription is messenger RNA. It's carrying the message of the protein. And then you have translation, which occurs in the cytosol, or you can use the word cytoplasm if you like to. And what we end up with is a protein. So having said that, let's look on page 104. And we're looking at the first column. You see it says, recall from earlier in the chapter that DNA is composed of a long chain of nucleotides containing genes. Those nucleotides are the genes, and they're set up in particular sequences. Actually, we can take some of those, and we're not going to get into all that. We can take a gene down, we can take parts out, and then we glue it together. How do they know to do that? So that you get the right sequence of nucleotides for a particular protein or a gene that's going to produce a protein. So you see in right after genes, the bold print term on page 104, column 1, you see it says, although there are only 4 nucleotides in DNA, there are 21 different amino acids, just like our alphabet, 26 letters. You come on down about 3 lines, you see how it says, however, there are enough combinations with groups of 3. You got to have groups of 3 nucleotides. And it is these groups, and they give you an example like AGT or CCG, that are words that code for amino acids. These words are called triplets. So the code is written in triplets. Each triplet represents a particular amino acid. Come down to the second paragraph. During transcription, it's copying the code. Each DNA triplet is transcribed into a complementary 3-nucleotide sequence on messenger RNA. We make this step by step. And each of those 3 nucleotides in messenger RNA is called a codon. Do not call it a condom, okay? Unless you just don't want any credit. So it's a codon. In essence, the codon is a complementary RNA copy of DNA triplets. And you do not have to know what I'm about to show you. But if you look below, you see a nice little cleared area there, and you see a box, and on the left side it says first base, and on the top it says second base, and on the right-hand side it says third base. So this figure has all the possible combinations, 64 of them, of 3 nucleotides. So you can see that UUU and UUC stand for the amino acid. You don't have to know these things. Phenylalanine, that's an amino acid. Come all the way down to where you see the green bar and you see AUG. And you see that means start. That's where the gene is going to start. If you look over in the second column, or excuse me, the fourth column, and I'm just doing this because you would recognize the letters, you see UGA, University of Georgia, go Dogs, right? That's where it stops. So we have a beginning point and a stop point. And all in between there represents the code for a protein. Now, having said that, and knowing that DNA is a polymer of nucleotides and they're arranged in threes or triplets, let's look over to page 106. We're looking at figure 329. So you see up above, at the first part, you see this double helix. You remember that DNA is a double helix. And I think we covered that back in chapter 2. So go back and review that. So here's our double helix. That's DNA. And you see they've marked out with the little brackets, gene. Now don't worry about promoters. There's a lot of things that goes on. We're not going to cover that, but just the bare bones here. Transcription factors, don't worry about them. This it gets deep and deep and deep and deeper. So anyway, that's the gene. Now remember, I said only one side of the DNA codes. We don't know why that happens. All we know is that one side of the DNA codes. But we can work with that. You don't know how your transmission works, but you know where park and reverse and drive are, and you can get to school or go to work, right? So there's the gene, and here comes this enzyme. Now, how do you know this enzyme in the next figure? It says RNA polymerase. How do you know that's an enzyme? ASE tells you it's an enzyme. And notice it says polymer. What do you think this enzyme is going to do? This enzyme is going to make a polymer, a chain of repeating units. And it happens to be an RNA polymer of RNA nucleotides. So you see it opens up where the gene is. And RNA polymerase opens up the gene, and there may be another enzyme involved, and we just don't know it yet. But come to the next figure. We're coming down to the third figure there, and you see that gene is open. You see the RNA polymerase, that kind of brown structure, and look, coming out of the DNA is this, not a double helix, it's a single strand. And you've got all these nucleotides hooked to each other. And so we're building an RNA transcript. It's called messenger RNA, carries the message of the protein. Look at the enlargement. Follow the little dotted lines down, and you see the purple bands of the DNA, and you see they're pulled apart. And then you see the red band, and that's the messenger RNA. So you see the little colored blocks in there that fit together, A and T and G and C and so forth. Those are the nucleotides. And so you have a cytosine nucleotide and a guanine nucleotide and so forth. And And then you see an adenine and thymine, cytosine and guanine and cytosine. And then look over here to a green one, you got a uracil. Remember that RNA does not use thymine. And we don't necessarily know why, but it plays that role. Uracil is only found in RNA. And you're going to actually have 3 types of RNA, as you'll see in a few minutes. So we're making this polymer of RNA from the DNA triplets. You can see the words in there, triplet. You can see the mRNA transcript or the mRNA, messenger RNA copy. And then you come down to the last little diagram and you see you've got this snaky looking thing that is called the messenger RNA transcript. Transcript, remember, is a copy. So now we have completed transcription. It's a code for a protein, and we have to decode it, read the code, so that it turns into, it doesn't turn into, but it guides a sequence of events to produce a polymer, which we're going to call a polypeptide, the primary structure of a protein. So you come over to 107, and you can look at flashbacks and so forth. You come down into the middle of there, and you see the bold print term translation. Translation takes place with the help of ribosomes. There's a second kind of RNA. We've got messenger RNA that has the code, ribosomal RNA that helps, and involves another type of RNA that acts as a molecular translator, named transfer RNA. So we actually have 3 types of RNA. Different sequences of nucleotides determines that they're going to be ribosomal RNA, messenger RNA, or transfer RNA. So you see it says, recall from the previous model that transfer RNA is manufactured within the nucleus. We talked about that a little bit earlier in another video, from a DNA template. It's called transfer RNA because it picks up amino acids and carries them, transfers them to the messenger RNA. This just blows me away. Look down at this little cross-looking thing, or some people say it's a 3-leaf clover or whatever. But you see it has, if you look at the bottom of that, where it says A, structure of transfer RNA with bound amino acid. See, one end is the anticodon, which reads the codon of the messenger RNA. You go up to where you see the end of the transfer RNA, that'd be the north end, I guess you'd say, and it's got PHE on it. So that transfer RNA picks up phenylalanine. It doesn't pick up glycine. It doesn't pick up methionine. Those are all different amino acids, which you do not have to know. But the point is, there's a specific part up there where it picks up phenylalanine. How does it know to do this? Sometimes all we can do is observe. So you see that area over there is the attachment area for amino acids. The south end, if you want to say that, or the bottom end, and the anticodon, that's going to hook up to a codon on the messenger RNA. Now, you're going to have to match up anticodons and codons. So what we have over here in the box on page 107, I want you to go through that. And you can look at the first box and you see amino acid, there's isoleucine. You don't have to know them, don't worry about knowing them. And look at transfer RNA. The anticodon is UAG. The messenger RNA codon that would attach, or I should say the transfer RNA attaches to the messenger RNA codon AUC. A and U match up, A and U match up again, and then G and C match up. Going down a little bit where you see the purple, that was derived from TAG, thymine, adenine, guanine. The thymine had to connect with adenine in messenger RNA. The adenine nucleotide can't attach to T, thymine, because messenger RNA doesn't use it. So you got uracil, and then G and C match up. So you can work out some of the codons, anticodons, and so forth in here, and that little work should help you understand this. So now we look back over to the left, and down at the bottom, you see this little transfer RNA molecule, part B, carrying phenylalanine, and then you see it has an anticodon of AAG. What would be the codon for this anticodon? Somebody might ask you a question like that. Let's go over to page 108. Page 108, we're looking at the ribosome down at the bottom. We're looking at figure 331. And you see here is the transfer RNA, got the MET on it. Don't worry about the names of the amino acids. But you see the U is opposite of A, the A on the transfer is opposite of U, and the C is opposite of G. Anticodon and codon. So 2 parts to the ribosome. Now we got all 3 of those RNAs together. You see to the left, you see the mRNA transcript. You see the, I don't know what you want to call that thing, a pounder. It looks kind of like something we've got in our kitchen when you can chop up onions. But anyway, that blue structure, it's got a long tail going up. That's going to be the transfer RNA with the anticodon. And then there's the amino acid. And then you got the big RNA around it, ribosomal RNA. In the next step, we have another transfer RNA aligned next to the first one, the P, which is phenylalanine, and A is alanine. P is not phenylalanine. That's the way they label them, and that's okay. You don't have to worry about labeling them. But you see EPA, so I guess that's access to the right, and I don't know what P is right now. I haven't looked it up, but E might be an exit. So anyway, you see you got 2 transfer RNAs next to each other, and you got 2 amino acids next to each other. And so they line up with their codons. The anticodons mesh with the codons. And then you come to part 2B. This will be the 3rd structure to the right. And you see there's a little bond there between methionine and glycine. That's a peptide bond. So we have a bond made. What chemical reaction took place? Look it up. I see that on the test. And so we move on over to page 109. And the first one that carried methionine is, we can say, kicked out, it leaves or whatever. And we have a P and an A in there. Again, methionine, glycine, alanine, and their anticodons are matching up with the codons. A opposite U, C opposite G, and so forth. That's how we build a long polypeptide. That'd be 50, 100 amino acids long or something like that. The ribosome is helping to make the bond between the amino acids, and you look over to the right and you can envision, as they say, a completed polypeptide. A completed polypeptide. And then there's always a codon there, an anticodon that, as it says, a release factor. You got the end of the line. We've even got codons and anticodons that tell you to start and to stop. But the point is, you've got yourself a polypeptide. Now remember, let's look over here on page 110. And you see the whole sequence there in one page. And so you can walk your way middly through there. And when you come to the bottom, then you see it says modified functional protein. And remember how we spoke of the ribosomes on the ER? They were involved in producing the polypeptide. How do you get the other levels? We have spoken about that. So make sure you know that. That's back in the ER in this same chapter. Okay, so that's where the secondary, tertiary, and quaternary changes occur. You just got to look it up. It's associated with the ER, but look it up. How does it happen? And I think you probably already know this, but you probably had it in lab, but you can look over on page 114 and 115. Remember those stages of mitosis? Interphase, of course, is a resting kind of state, and they've got some other activities going on in that interphase state. But in terms of mitosis, you got those 4 stages, and interphase is a resting stage in between. Okay, I think we've covered everything. Let me make sure that we have done this. Well, I think we've done that, I believe. Yeah, we've done that. This back in chapter 2, where we talked about nucleotides and DNA and RNA. All right, I'm going to conclude this lecture, and if you have any questions, you know how to reach me. I'll probably be tied up this weekend. Again, like I usually am, have some other responsibilities. You can shoot me an email, but I won't get to it till Monday. Okay. So next Thursday is when we will have our first exam. It'll start at 8 o'clock in the morning and go to 6 o'clock on Saturday night. So you'll have all day, 24 hours on Thursday, 24 hours on Friday, and then what? Till 6 o'clock, almost 24 hours on Saturday. About 12 hours. So you got 2.5 days to do that. All right. Do you have any? Well, you can't say anything at this point. You guys, some of you are probably still asleep. Anyway, I'm going to close out of here, and I'm also going to ship you a Word document. Have a good weekend.